Very good morning, students. So in this video, we are going to discuss about the plant tissue culture technique, call it as micropropagation. Let me share that PPT to start the lecture. Okay, so micropropagation, we call it as in vivo clonal propagation of plants. It is tedious, expensive, and frequently unsuccessful. So that's why in vitro clonal propagation through tissue culture is referred to as micropropagation. This technique was first started by Morin in 1960 when he was propagating orchids, and now it is applied to several plants with some modification. Micropropagation is handy technique for rapid multiplication of plants in plant tissue culture. So moving on to the advantages of micropropagation, faster multiplication. So you can able to multiply large number of plants in a short period, and sexually derived sterile seeds can be propagated, and raised plants from the plant tissue culture of micropropagation technique pass through an undesirable juvenile phase, which is avoided in the asexual reproduction. Gene banks can be more easily established by cloning propagated plants. So these are the advantages of micropropagation. Otherwise, we can call it as clonal propagation. So moving on to the stages of micropropagation. So we have 5 stages. Stage 0, selection of mother plant and its maintenance. So to take the explant, the mother plant in stage 0, you have to select it in 15 days old sapling. You have to grow it under the greenhouse condition. Stage 1, you have to take the explant and you have to start your initiation and establishment of culture. Stage 2, after incubation, proliferation of shoots. Stage 3, proliferation of roots. Stage 4, after you got a complete disease-free plantlet, you can go for acclimatization or hardening process. That is stage 4. So moving on to the techniques involved in micropropagation, multiplication by axillary buds or apical shoots or meristem tip, or multiplication by adventitious shoots. So let me see multiplication by axillary bud. Bud in the axillary region are highly meristematic in nature and disease-free. So you have to cut the shoot apex, which is disease-free. As usual, you have to do the sterilization procedure, how we are following the aseptic techniques to initiate a culture in a lab. So stage 1, you are inoculating. Stage 2, shooting will be there. So the plant hormones, high cytokinin content, low oxygen level will promote the shoot. Low oxygen, high cytokinin content will promote the root. Then you have to go for hardening the plant. So this technique of micropropagation is by using axillary buds or apical shoot as your explant. And the second technique is the nodal culture or bud culture. Buds in the nodal region of any plant also, you can cut it as an explant and you can start the micropropagation. So buds are nothing but the dormant. So if you want to show the bud culture, you have to cut off the meristem tip. So once a meristem tip is keep on growing, it won't allow the bud, dormant bud to grow. So when you are using explant, the bud one, meristem tip, you have to manually remove off. This is the another technique by using bud in the nodes in micropropagation technique. Bud can be used from the elongate plant as well as the rosette plant also. So based on the habit, wherever it is bud is there, you can aseptically excise it and you can initiate the bud culture in micropropagation technique. And the second technique, that is multiplication by adventitious roots. Let me tell you what do you mean by adventitious roots. Stem or leaf structure that are naturally formed on plant tissue located in sites other than the normal leaf axis, we call it as adventitious shoots. Example, bulbs, tumors, rhizomes. So these and all, other than its normal axis, it can form on any other places in the plant body. That also you can excise and you can start the micropropagation technique. And these are this you might have experienced, I guess. Okay, students. So from this video, I hope you understood what is meant by micropropagation, importance of micropropagation, advantages of micropropagation, the techniques involved in it. You can use the meristem tip as an explant to initiate your plant tissue culture, and buds in the axis, both in the elongate plant and in the rosetti plant. You can take it as an explant to initiate the culture. Then how adventitious shoots and roots you can cut aseptically, and you can start your micropropagation technique. And the stages involved in it, it has to start from stage 0, maintenance of the mother plant, then stage 1, initiation, 2 and 3, proliferation start, and finally in stage 4, you will get aseptically disease-free plant if you have done it properly. Thank you.